Dr. Schmid, thank you so much for joining us today. We're going to be asking you some questions to better understand the courses you teach, the investigations you conduct, and the thesis project you supervise with our fourth-year honors specialization students. So could you tell us a little bit about yourself?
I started at Western about 15 years ago as assistant professor. I am now Vice Dean for Basic Medical Sciences here at Schulich. I'm a neuroscientist, which is a very interdisciplinary discipline. My background is biology. I studied biology in Germany, and before I came to Western, I built a career in Germany, and then I had an opportunity to come to Western. That's why I'm here.
And could you tell us briefly what you're currently investigating?
Yeah, we are focused, well, I have two major arms of research. One is to figure out the mechanisms in the brain how we filter sensory information. And the idea behind that is that any input into our brain is sensory information, coming from our five senses or from a huge number of internal sensors that we have in our body. And that information reaches the brain all the time. And only a very small fraction of that will become aware. And so there are built-in mechanisms in the brain that filter out unimportant information or information we are not supposed to become aware of. And we look at these mechanisms of filtering sensory information. And the second arm is a more health-related one, and that is looking into disorders and diseases where this sensory filtering and sensory processing is not working properly. And we focus on neurodevelopmental disorders like autism, where people are easily overwhelmed by the abundance of sensory information because these very basic filtering mechanisms don't work properly. So we look at basic sensory information processing, mostly in the brainstem, because we use animal models of neurodevelopmental disorders. And it is these old areas of the brain, like the brainstem, that are very similar between humans and, for instance, rodents. And so this is why we focus on these areas. We think they are important, but we also appreciate that these are the most conserved part of our brains.
So Dr. Schmid, what are the most commonly or most interesting use pieces of equipment in your lab?
Yeah, I don't think there's a single one, most used one, because I think the times where researchers just would focus on one method, these are over. We are very interdisciplinary and multi-approach nowadays, but we are here in the wet lab, and within this lab, I think the most used equipment is, for once, a tissue slicer, a cryotome, where we slice brains post-mortem. So whatever animal model we use, we take the brains out afterwards, and then we can slice them, and then we do histology and look what's changed in these brains. So the brain would be mounted here and frozen, and we would slice small slices off that would stick here, and then we take a brush and take it off and put it into a petri dish. And this is how we get sections for histology from rat brains. And the other one, which is a very special piece of equipment, is in a small room to the side, which are our electrophysiological rigs. So this is where we perform patch clamp electrophysiological recording. So we record from single neurons through electrodes, and these neurons are in brain slices, acute brain slices, so they're still alive, and we can record action potentials and postsynaptic potentials and things like that.
So Dr. Schmid, now that we are in the SSC, which is a different part of your lab, could you let us know what this piece of equipment is?
So this is one of our soundproof boxes. Since we study processing of sound in most cases, we need to control the environment, so we need to have a very quiet environment. And so the soundproof boxes are padded on the inside. It's very quiet inside. There's really no background noise. And inside these boxes, we do things like recording ultrasonic vocalizations of red pups, but we also do electrophysiological recordings of auditory brainstem responses to sound within these soundproof boxes.
So can you tell us what the equipment over here is?
So these are our startle boxes. We measure the startle response of rats in response to a sudden loud noise. We know from kids with autism that they startle much higher than other kids, and so the same is true for some animal models that we have. So we put a rat into this tube here, which is pretty similar to the tubes that they have in the cage where they like to sleep in. And then we put these rats onto a motion-sensitive platform inside the box. The entire equipment is run by a computer and is soundproof. And so when the rat sits in there will be some random, sudden, relatively loud noises, and the rats startle, and these motion-sensitive platforms then measure the amount of startle. It's digitized and directly stored in a computer. And we can measure 4 rats at the time. Startle boxes.
So which courses do you currently teach or have taught before?
So because of my role as a vice dean, I don't teach that much anymore, but I do still teach the second half of a half course, Neuro 2000, which is on sensory organs and sensory systems. I also teach in the third year histology course, anatomy and cell biology, where I teach 8 lectures about the brain and the spinal cord and the peripheral nervous system, as well as on the eye and the ear. I originally created a 4th year neuroscience course, integrative neuroscience, that I used to teach, but I don't teach it anymore. Now I only teach a very small section about learning and memory in this course, in this 4th year course. And I do teach also a small section in a neuroscience graduate course. That's all the teaching.
So if there's one thing you could tell students looking to excel in your classes, what would you say?
Excel in my classes. I think what I normally like, or the students, that stand out, are the students that are asking a lot of questions, smart questions, like interesting questions. I think that is one way for students to really stand out in classes.
And what are the top 3 things you look for in a prospective thesis student?
That is interesting. It's always a package that comes. For me, important is a real interest in doing research, an attitude that that a student applies him or herself. But I do like students that also have a good work-life balance on the other hand. So work hard, play hard is a type that I personally like.
And what would be the most challenging part of your research?
Well, I think by far the most challenging part of the research nowadays is the funding, is to arrange for research grants and get funding so that we can actually pay the graduate students and undergraduate students to do the research, and for the animals and all the other things that we need to perform research.
And my final question to you is, what is your favorite type of coffee, and when was the last time you broke something in your lab?
Coffee, I'm not a big coffee drinker. I drink any coffee as long as it doesn't contain any artificial flavors. So I'm very good with the Tim Hortons. The last time I broke something, I haven't broken anything in a long time. That's mostly because I am not much hands-on anymore myself in the lab. I'm constantly repairing equipment that my students break. That is just something that happens. It's unfortunate, but it's sometimes unavoidable. So I'm actually the one who fixes things. I try not to break anything.
So thank you so much for your time and your insight of your lab today.